Hey, this is Joel Duff, and I have some further thoughts. On comments that I made on a live stream last night. And in the live stream comments, I was talking about horizontal gene transfer, and in particular, I was talking about a form of horizontal gene transfer in which pieces of mitochondrial DNA genomes are transferred to the nucleus and become incorporated in the nuclear genome. This has a name, NUMTs, and it stands for nuclear mitochondrial DNA. And what I want to do here is just go to literature and show some examples and potentially the importance of NUMTs and genome evolution. So we have that coming up. Yeah, so what inspired me to decide I needed to talk a little bit about NUMTs today, nuclear mitochondrial DNA. It all has to do with my watching a live stream last night on a station called Examining Evolution. And there, Sal Cordova was having a great conversation with Rebecca of Bread of Life, and they were discussing problems with eukaryotic evolution. Right? Eukaryotes are cells that have nuclei in them, as opposed to prokaryotes, which are cells that lack nuclei. But in addition to having nuclei, eukaryotes have subcellular organelles, for example, mitochondria, right, the power plant. And that mitochondria has its own little genome, but that genome doesn't do nearly enough to run a mitochondria. It needs a lot of assistance from the nucleus. And so in this little diagram here, we have you've got the nucleus of a cell, and inside that, right, you've got your chromosomes, which have your different genes, right, at various loci on those chromosomes. In order to run your mitochondrion properly, you have to make products from those genes. You have to export them, and it's showing two different types of things that come out of there. But let's look at nuclear encoded proteins. Those proteins have to be exported and then imported into the mitochondrion. And here we're indicating about 1,500 different proteins, right, imported into the mitochondria, where they then work with a much smaller set of proteins and other non-coding RNAs that are encoded in the mitochondrial genome, right? The mitochondria has its own genome in animals and in plants. And Sal was, properly talking about just how incredibly complex this is, right? This is a very delicate dance of molecular biology where you have these large sets of genes and processes that occur in the nucleus that control the mitochondria. And then in the comments section, of course, what comes up is a question about, well, how exactly then did this really complex system evolve? Like, where did mitochondria come from? And so the question was asked is, what did Sal think of endosymbiotic theory? And endosymbiotic theory is the proposal that a long time ago, a eukaryotic cell, or a very simple early eukaryotic cell, engulfed a bacteria and made it subservient to it, right? Or maybe the bacteria was parasitic on it, but lived inside that cell. That bacteria originally would have had a whole genome that probably had 1 million base pairs and several thousand genes. But today, the mitochondria in human beings and in other animals is very, very small, only 16,569 base pairs for a typical person. Plants have much larger mitochondrial genomes, but nothing like the size of a bacteria. And they do have a small set of genes. So where did all the genes go if they started out as a bacteria? Well, the proposal is that many genes from the original bacteria that lived inside of cells transferred those genes into the nucleus, where they then reside, and the nucleus then uses those genes and then controls the mitochondria from there. All right, so it's a transfer. I guess you could call this a transfer of information from what was the bacterial genome into the host cell, all right, into the host genome, the nucleus. And now the host cell now controls that subservient organelle, the mitochondrion. And the mitochondrion does have its own little small instruction set to sort of like take care of a few of the operations that it needs to. One of the theories is that some of those genes have difficulty exporting and importing through nuclear membranes and mitochondrial membranes, and so it would be difficult to transfer that information to the nucleus. Better just to store it in the mitochondria and bring it up when you need it right there. All right, so all of this precipitated this idea that okay, so maybe mitochondria just represent old bacteria. But Sal was pointing out that this is really complex. All right, there's so many hundreds of different genes in the nucleus, and this would require some kind of how would you get those genes into the mitochondria? And I suggested in the comment section, I said, horizontal gene transfer, right? Which is what I just described, right? Genes being transferred into the nucleus, as and then the conversation got sidetracked, and I got sidetracked, right? So because Sal was wondering about, I'd suggested that genes could be moved over. He thought that was probably a fairly difficult process in order to get them. What would be the mechanism of doing that? I think he'd even asked me, what is the mechanism? Has that actually been seen? And all I could say at the time in the chat section, in my very limited number of characters, is that I believe that there was a strong inference that genes had been moved in the past to the nucleus, but that we hadn't physically actually seen that happen. But that got me going on. As I sat and listened, I also was doing a Google Scholar search and reading a number of papers, and that continued on into this morning into what I'm going to show you now. So Sal, this is actually for you, Sal. You're wondering about how this might happen, or whether it does happen, whether genes can, or pieces of DNA can be transferred fairly readily from one organelle to another. And I want to try to show you that there's very compelling evidence that it can happen. Now, this is just a small subset of the things that you talked about, and it certainly doesn't explain all the types of questions you were raising in that live stream. And I'm not at all attempting to do that. I'm just taking this one little tiny topic and of horizontal gene transfer and trying to show that it is something that does happen. And then once you know that it happens, the question becomes, is it a significant thing? All right, can it significantly shape organisms over time? And there you can have a variety of different arguments, statistical arguments and so forth, about probabilities of that being a significant evolutionary, having an evolutionary function over long periods of time. But I just want to show that horizontal gene transfer is a real thing, and show you some examples. Okay, so where am I going from here? Let's move right to our examples. So I want to let's just start with human beings, because we're always most interested in ourselves and what's happening inside of our own genomes. And what I found fascinating, and I had forgotten about this, I knew about NUMTs, and I actually have talked about them in class before many, many years ago, but I hadn't really done any recent research. And of course, with all the genomes that we're pumping out, we now have a much greater capacity to identify specific segments of DNA that are in genomes, and find their actual endpoints where they attach to. If it's a foreign piece of DNA, how is that attached to the genome of the host organism? And we're discovering that there are way more pieces of mitochondrial DNA that are in our nuclear DNA, and not just for human beings, but for lots of different organisms. This kind of movement of pieces of genomes from one organelle to another, as wild as it sounds, because it just doesn't sound like it would be easy to do. It's hard to imagine the mechanism in which this would happen, and yet it clearly does happen, as I will hopefully show in a moment. So let's start out with this paper first in Forensic Science International Genetics by Marshall and Parson, interpreting NUMTs in foreign and forensic genetics. So forensic genetics would be those who are using DNA to identify ancestry, usually, or in the case of criminal cases, to use DNA sequences to identify, as specifically as possible, individuals that might have been involved in a crime. And of course, a big concern in forensics is falsely identifying somebody, right? Falsely accusing somebody based on DNA. So there can be contamination of DNA, resulting in incorrect evaluations. But what gets more interesting here, and the reason I wanted to start with this example, is that it turns out that there are, and I'd heard of cases like this, but I didn't realize the extent of it. There are cases of individuals who have more than one copy of a mitochondrial genome. All right, so we think of our mitochondrial genome as, remember, we got our cell, we have our nucleus, all right, our nucleus, and then we have our mitochondria. And in our mitochondria, we actually have multiple copies of our mitochondrial genome. But if you were to look at each copy, they should be pretty much the same as each other. So we'd say you have a version of your mitochondrial genome. And that whole ancestry thing, looking at our female lineage, all right, is tracking back the mitochondrial genome, which we get passed from female to female through history. And so there would be one lineage, and I've had my genome tested, and I know what my mitochondrial DNA is. Well, it turns out that some people have had their DNA tested, and they have conflicting data because they have two types of mitochondrial DNA that might be very different from one another. And initially this was kind of a puzzle. How could this be? It's contamination of the sample and so forth. Well, what we've discovered is that there are individuals that have a copy of their mitochondrial DNA embedded in, or a copy of a mitochondrial DNA embedded in their nuclear DNA. So they have a mitochondrial DNA and they have a nuclear mitochondrial DNA, a NUMT. And this nuclear mitochondrial DNA isn't necessarily the same as their mitochondrial DNA because they've inherited their nucleus, right, potentially from their father, all right, or this particular portion of the chromosome from their father rather than from their mother. And that's a different lineage. And it could have happened long ago, so they've been passing this mitochondrial DNA down in their nucleus over and over and over again through time, through many generations. And that becomes separate from the ancestry of the female line of the mitochondrion. And so you can have two different mitochondrial genomes. And so if you're doing ancestry analysis, the ancestry analysis can be confused because if you don't understand that which of the genomes you're looking at in your analysis, you could actually be tracking the male line or the chromosomal lineage versus nuclear chromosomal lineage versus the mitochondrial lineage. And so it was these weird cases that raised the question of like, how could this be? And that's where they found, and now of course now we're doing whole genome sequences, we can now just scan the whole genome and find these chunks of mitochondrial DNA that are inside of human nuclear DNA. So as they say here, nuclear mitochondrial DNA segments, NUMTs, were discovered shortly after sequencing the first human mitochondrial genome. So once we had the sequence of the mitochondrial genome and we started sequencing nuclear DNA, we started seeing little pieces of things that look like mitochondrial DNA. And at first this was kind of mysterious and usually was tossed aside as we've got a sequence read problem and so forth, or contamination, because it really is hard to imagine how mitochondrial DNA would be incorporated, would be communicated and passed into the nucleus. So now what this paper covers is they're talking about what is the frequency of this. Now that we've done a lot of human genomes, how many people are carrying around pieces of mitochondrial DNA in their nuclear DNA? Well, the answer is we all are, right? Every single one of us has numerous portions of mitochondrial DNAs found in our nuclear DNAs. Now I want to stop here for a moment because I know Sal would have maybe this question. And you just say, well, how do you know it's mitochondrial DNA? Yes, it might have sequence similarity, but there could be segments of nuclear genome that just happen to have nuclear similarity to those genes in the mitochondria because there are maybe similar genes in the nucleus. All right, the similarity is very, very high, so in many cases 100%, but not all cases. There are differences between the mitochondrial sequence in the mitochondria versus in the nucleus. I'm going to get to in the next paper why I think that I think a piece of data that will convince you that DNA is actually being transferred from the mitochondria and inserted into the nucleus. Here is, so look at my blue box here. For example, a study of the 1,000 Genome Project data. So the 1,000 Genome Project data is sort of this initial really good set of 1,000 human genomes presumably representing a good set of diversity of humans. And what they did was they searched that, and they discovered 141 new NUMTs they identified, ranging from 39 base pairs, so just 39 little base pairs, but those base pairs have sequence similarity to the mitochondrial genome, not to anything else in the known nuclear genome, up to 16,000 base pairs, an entire 16,000 base pair piece of mitochondrial DNA. Well, that's actually approximately the size of the mitochondrial genome itself. And they added that to the established catalog of 755 NUMTs already known, already identified prior to scanning more genomes. So just a few initial genomes immediately discovered a bunch of things. But now we've done 1,000 genomes, and of course now we've looked at a lot more genomes since. We're discovering thousands and literally thousands of these little fragments of mitochondrial DNA in nuclear DNA. Prior to this, only a small fraction of NUMTs were believed to represent recent insertion. So that's another question we have is, when did they get inserted? If they really are pieces of mitochondria that were transferred to the nucleus, when did that happen? Is it happening right now, today? Did it happen in the last few hundred years or last few generations? Or are these really old events that we're talking about? The answer is it's all the above. So phylogenetic analysis of observed nuclear inserts, including new ones in non-coding control regions, however, suggests that most of the NUMTs have been inserted within the past 100,000 years. The reason they're saying 100,000 years is, that's the conventional, that's the conventional ancestry analysis of humans, is that they're about 100,000 years old. And the reason they're saying that it looks like they're within the past 100,000 years is because not everybody has all of these. So if you look at a broad spectrum of genomes now and you say, like, okay, I've got a particular segment of 5,000 base pairs of mitochondrial DNA on chromosome number 2 at X position, and I might find that there's a lot of other individuals that also have that at exactly the same spot, especially people that are related to me, right, in my particular lineage. But not everyone in the world has that particular thing. So it looks like somewhere during human history there was a horizontal gene transfer event where a piece of mitochondrial DNA was transferred into the nucleus in someone, and they then gave that to their children, who have then been sharing it right with their offspring after that, such that we have some individuals that have that share that particular site. So that'd be a shared derived trait, a synapomorphy, we call it, and in phylogenetic parlance. And then, let's go on here. Furthermore, over 50% of NUMTs identified show very low frequencies across tested samples, which indicate these NUMTs are young and the process of NUMT insertions into the nuclear genome is still ongoing. So 50% of the many hundreds of NUMTs that have been identified are only identified now that we've seen and we've looked at a lot more genomes. The reason we didn't see them before is because some of them are quite rare. They're only found in a few individuals potentially, or possibly a single individual, as we'll see in my next paper. All right, so all of this suggests that NUMTs are constantly being fed from the mitochondrial genome into the nuclear genome, and they're actually expanding our nuclear genome because they're adding pieces to the nuclear genome, making it larger. So there is selectional pressure to reduce the size of genomes, but there's also pieces of stuff that are being added to our genomes as well. In this case, from the from the mitochondrial DNA. In plants, it's even more pressure because they have a lot of pieces of their chloroplast and pieces of their mitochondrial DNA in their genomes, and some of them have vast amounts. Now, not only do we have copies of pieces of mitochondrial DNA in our genomes, but some individuals have replicated those or duplicated them through mutation again as we copy our genomes, and made multiple copies of that piece of mitochondrial DNA. And so the last point here is that the substitution rate for the rate at which insertions happen into our genomes is kind of similar to the rate at which our nucleotides are changing, our single nucleotide polymorphisms are accumulating over time. So that's really interesting in itself. I just want to also point your attention up here to this other blue box, right? Mega NUMTs. All right, that's just the name for the really large pieces of mitochondrial DNA, not just small little snippets. They looked at 7 of 11,000 trios. All right, so you're looking at mother and father and child. And they found a number of different NUMTs that occurred only in those individuals, so show it's passed down. And these are really large. So some of these, in some cases, they're about the size of an entire mitochondrial genome. So the entire mitochondrial genome is copied. And that brings me back to why I started with this particular paper, right? I said that if you're doing some kind of forensic analysis and you're trying to identify the specific type of mitochondrial DNA this individual has, and you're just using some kind of molecular marker that goes in and tries to copy out a piece of the mitochondrial DNA in order to sequence it and get your molecular profile or your fingerprint, some people's fingerprints are the wrong fingerprint because they're copying out the section from their nucleus. And they didn't know it originally. And this is why this is the importance of this kind of work, right? They're making people aware that this is a type of error that can occur. We're finding that you might actually be copying a nuclear version of your mitochondrial genome versus your mitochondrial genome. And since your nuclear mitochondrial, getting confusing, right? I have to keep saying these same words over and over. Your nuclear mitochondrial DNA, right? Your NUMT, you might have inherited that from 50 generations before, which is completely unrelated to your mitochondrial DNA that you got from your maternal line. And so you can have very conflicting results in terms of your ancestry based on those two things. So the message of this paper pretty much is you have to be really careful to make sure you're actually reading and looking at the mitochondrial DNA. And there are ways to separate mitochondria and then extract DNA from mitochondria and so forth to double check these types of things. All right, so that paper I think illustrates that within humans, you have multiple different copies and pieces of mitochondrial DNA that have been inserted at different times in human history. Now, what I didn't say before, I'll wait. We'll get to it. Because I wanna stress one other thing first before we look at the broader picture of NUMTs across, say, mammals, and even beyond mammals. What about this paper here? A frequent somatic transfer of mitochondrial DNA into the nuclear genome of human cancer cells. So what I liked about this paper is that it shows that in one single individual, right? One individual, they started out, right, with one copy of their mitochondrial genome. If you looked at their parents, they would have had, the mom had a mitochondrial DNA, and then you could look at the individual early on and see that, okay, you got one mitochondrial DNA. And your nuclear DNA it has some pieces of mitochondrial DNA, but they've been inherited from hundreds of generations earlier. But in this paper, what they're saying is, these individuals, in multiple different cases, looking at cancer cells, which is what they were interested in, they found that large pieces of the mitochondrial DNA of that individual had been inserted in their nuclear genome. And how did they know that? Because they compared cancer cells with non-cancerous cells inside the same person. And so you're essentially looking at what's your ancestral state. Your ancestral condition is you just have the normal set of NUMTs, right, that most of us share. But then in these cancer cells, we see there's a bunch of extra NUMTs in the cancer cells. Those have to, going back to asking, have we actually seen it happen? Well, even in this case, we didn't actually see it happen. But I don't know how to explain it other than miracle, right? That they were just created in the cancer cells. It seems pretty obvious to me that mitochondrial DNA is being transferred into the nucleus actively in this particular individual, actually these individuals that are mentioned in this paper. And so somehow it's happening, right? There is transfer. Now they actually do talk about the types of junctions that they find and where the mitochondrial DNA is found in the genome, because they can do highly precise DNA sequencing and they can identify exact locations where these mitochondrial DNAs are spliced in compared to the normal cells outside of the cancer. And they point out that most occur in conjunction with internuclear genomic rearrangements. That's where DNA itself is breaking and reforming, because there are splice sites in DNA cells. Even as DNA copies itself and undergoes mitosis, there are places that have breaks and repair. All right, those spots that are most likely for that to happen, there are hot spots in the genome where this breakage is allowed to occur. Features fusion fragments indicate that non-homologous end joining. That's just the fancy name for the molecular mechanism that's doing this genetic exchange, and replication-dependent DNA double strand break repair. Right, that's the type of repair that after you break your DNA and you put it back together again, there's a process by which you basically fuse it back together to hopefully make it new again. Are the dominant mechanisms involved. Remarkably, mitochondrial nuclear genome fusions occur at a similar rate per base pair of DNA as interchromosomal nuclear rearrangements. So if you look at the amount of rearrangement that occurs in cells in mitosis, because we actually do undergo some rearrangement of our genome as we copy it over time, errors are made in genome arrangement. That amount of rearrangement is similar to the amount of new insertions of mitochondrial DNA. And the last thing I want to point out down here is transmission of mitochondrial DNA to the nuclear genome occurs in neoplastically transformed cells, but we do not exclude the possibility that some mitochondrial DNA fusions observed in cancer occurred years early in normal somatic cells. What we're saying is we see that this is definitely happening in cancer cells, but we can't exclude the fact that transfer of mitochondrial DNA into the nucleus was occurring even in somatic cells that weren't cancerous. And then those cells happen to then become cancer, and then now we see it continuing in that particular cancerous cell line. So how do we accumulate new NUMTs over time? If we find a family group that shares a particular NUMT and nobody else among all humans have it, there's probably an origin point. What's the origin point? Well, somewhere in their cells, probably in someone's in their germline, right, making the sperm or egg. When they're doing that, maybe during that process there is that transfer. It could also happen very early in someone's life, in very early after the first cell, and as you're dividing cells, if you then had that transfer event happen there, then a good portion of your body might contain a new NUMT, right? And if that NUMT includes the sex cells, right, the sex organs, then those sex organs are going to produce sex cells that then have that particular new integrated version, and then they're going to pass it down from one generation to another, and therefore that is, you could consider that the origin of new information right there, because you've inserted a piece of DNA into a place that wasn't there before, and now going forward, individuals in the future will have that fragment of DNA. Now, the more interesting question, of course, is can individuals use that? And in most cases, right, 99.9999% of the time, this is going to be, I'm going to use the word, junk DNA, all right? This truly is fragments of a genome from your mitochondria put into your nucleus, but your nucleus doesn't know what to do with it in most cases, or the gene isn't even the full version of the gene, it's just a piece of the gene. That to me really is refuse, right? And this is part of the buildup of pieces of DNA in our genome that aren't used, all right, for any explicit process that we might do. But of course, if you insert your entire mitochondrial genome into your nuclear DNA, which some of us do have, we have an entire copy, or almost an entire copy, then that means you also have entire copies of all the genes. And if mutations occur that change the regulatory part of the upstream of those genes, it is possible that the nuclear genome will express those genes. Then, of course, you have to wonder, well, even if you could express the genes, can you use them? There are examples, and I'll just have a couple at the end, where we think that genes have been moved, fragments of DNA have been moved from one organelle to another, and then the nuclear genome has found a way to make it into a functional form of DNA. All right, now let's step back and we're going to start to broaden out now. And we're going to ask, where do we see NUMTs, and what does this mean for the bigger picture of how that might affect how genomes change over time? So what I'm showing you here now is the presence of different NUMTs in this paper. Well, here's this paper here from 2019 by Robert et al. Archaic Mitochondrial DNA inserts in modern-day nuclear genomes. All right, so archaic mitochondrial DNA inserts, all they're suggesting in that language is this is an insertion of mitochondrial DNA into nuclear genomes that happened a long time ago, right? It's archaic. And the reason we see them today is because they have been passed from generation to generation to us. And so what they're showing here is a series of phylogenies in which they've taken what you're looking at here is the actual NUMT sequence. So it might only be several hundred base pairs, or maybe it's 1,000 base pairs or 2,000 base pairs. That's the insert. And then they're looking at where that's found. And so let's just look at a couple of these. So up here at B, what we find is here's NUMT 23389. It's just the name of it, right? There's hundreds of these things, and they're just analyzing some in comparing it to other genomes of other hominids. So you have the Denisovans, right? And you have the Cimo de los Huesos genome sample. And those two do not have this particular sequence in it. But all modern humans and Neanderthals that have been tested all have this particular NUMT. Right? So the idea is that somewhere before all modern humans and Neanderthals, wherever they shared a common ancestor, all right, that gave rise to humans and Neanderthals, there was this insertion point. But after Denisovans and Cimo de los Huesos individuals. So that's where you have this new piece of DNA, new piece of mitochondrial DNA, which is inserted into the genome. And now you have it, I have it, Rebecca has it, all of my friends have this particular fragment of mitochondrial DNA that we would explain. The reason we would explain why we all have it is because we've all shared it from a common ancestor. All right, so this is very similar to the story of ERVs, I think you're saying, right? Endogenous retroviruses, because they're also inserted portions of fragments of DNA and jumping genes, right? And NUMTs are just jumping pieces of DNA as well. They're just a different type and they jump in a different way. All right, so then if we look at over here, we see NUMT4, 1787. Well, that's found in Denisovans, right? All the Denisovan samples have that particular piece, but modern humans and Neanderthals don't have that particular NUMT. So that suggests that in the independent lineage or separate lineage that gives rise to the Denisovans, their ancestor had a insertion of a fragment of their mitochondrial DNA into their nucleus. Down here we have NUMT3 1384, and it's found in Denisovans as well. All right, but Cimo de los Huesos doesn't have it, Neanderthals don't have it, and then all other living humans don't have that one either. Over here we have NUMT HS37D5, and it turns out modern humans, all of us share that particular NUMT, right? Every single one of us. But Neanderthals don't have it, all right? So it's suggesting it is a later derivation from after the common ancestor of Neanderthals, Cimo de los Huesos, and the Denisovans. And you're getting the pattern, right? Why are they not all the same? Because they're all independent insertions that happened at different times. And here's the great thing. All right, here's the really interesting thing is there's hundreds of these. And so if you start to track more and more and more and more of these, each one of them becomes a data point, and you can actually use the insertions, presence and absence of these in different individuals in different hominids to reconstruct a phylogeny of hominids. And that phylogeny is basically the same as a phylogeny as if you sequence some DNA sequence and do comparisons. So it tracks the same history. And this is true for lots of other different groups. The most famous NUMT examples are like cetaceans, where there's hundreds of different cetacean fragments that have been inserted at different times, and you can use just the insertions to make a phylogeny, a history of cetaceans, and that history is almost completely synchronous in line with the phylogeny based on other forms of data. All right, so let's just look at some other examples. And some of these I'm not going to go into any detail, I just was trying to find examples. So there's a paper here from 2017, striking pseudonymization in avian phylogenetics, meaning avians or birds have lots and lots and lots and lots of extra genes in them, or genes that are pseudogenes, so genes are not functional in those particular birds. And in particular, NUMTs are large and common in falcons. There are large fragments of mitochondrial DNA, in this case, 3/4 of the mitochondrial DNA of a falcon is inserted into the nuclear DNA of this particular falcon. This phylogeny over here shows that there are NUMTs, and these are different NUMTs in different places that have been inserted during the history of different falcon forms. In some cases, only one species has that NUMT, and all individuals in that species may have it, although, I mean, obviously not every individual has been tested, but you can infer that from the selection they've done. In some cases, just a couple species have it. And so if just a few species have it, well, that suggests that those species have a common ancestor that is sharing that NUMT. What about cats? All right. A recent transfer and tandem amplification of mitochondrial DNA into the nuclear genome of the domesticated cat. And what I found really interesting about this one is, well, let's just read the abstract here. The mitochondrial DNA of plants and animal cells is transcriptionally active genome that traces its origins to symbiotic infection of eukaryotic cells by bacterial progenitors. Right, so that's the endosymbiotic theory, and they're just taking that as like that is the consensus of the scientific community. As prescribed by the serial endosymbiotic theory, symbiotic organelles have gradually transferred their genes to the eukaryotic genome, producing a functional interaction of nuclear and mitochondrial genes in organelle function. So that's what we were talking about at the very beginning, right? That the nucleus is now the control center. It has, you think of it this way, it has taken, swiped, or gained somehow the genetic information for the bacteria and put it into the nucleus. And now the nucleus does the job of actually making many of those products and then sort of sharing them with the mitochondria. They also use them for other purposes too, some of those. And so the idea is that mitochondria have been shrinking over time, and they've kind of gotten down to sort of a minimum size level, right? They can't shrink much more. The reason we say that is because almost all animals have a similar size mitochondrial genome now. So that suggests that, and they have the similar set of genes in them. So those genes are conserved, right? You can't mess around with them as easily. But here we have in cats, we report here a recent remarkable transposition of 7.9 kb, so that's 7,900 base pairs of DNA, of a typically 17,000 mitochondrial genome. In other words, cats or felines have about a 17,000 base pair mitochondrial genome. You have a 16,500 something. To a specific nuclear chromosomal position in the domesticated cat. The integrated segment has subsequently become amplified 38 to 76 times and now occurs as a tandem repeat. So in the nuclear genome on one chromosome, not only is there 7,900 base pairs of mitochondrial DNA, which matches almost exactly the version of the mitochondria in the cat's mitochondria, but that 7,900 base pair fragment has been copied over and over and over again tandemly, so it means makes a copy of it and puts it next to it, 38 to 76 times. So different cats actually have different numbers of copies of these pseudogenes. They're called pseudogenes because they don't seem to have any function in the cat nuclear genome. They function in the mitochondrial genome. They're just sitting there in the nuclear genome and being passed from one cat to another. And then as they do that, they sometimes make mistakes and they end up repeating the copies. And so, cats, domesticated cats, are clearly derived from other species of felines. And other species of felines don't have this particular insertion of this mitochondrial DNA into their nuclear genome. And so once again, that's why I'd say this is I said in the chat, I said it's an inference. Yes, it's an inference that we look at the data and we infer that this transfer has happened. I didn't actually see it happen, but it's a really strong inference, right? The domesticated cats have something that their ancestor, that everyone agrees is their ancestor, doesn't have. And so they have up to, in this case, 150 some thousand base pairs of extra DNA in their nuclear genome. That's all copies of mitochondrial DNA. They propose a couple models for that. Basically propose a system in which the mitochondrial DNA, when copying itself, creates a closed loop, which is a subset of its original genome, which gets transported out of the mitochondria into then the nucleus, where either this thing gets copied over and over and over again, forms basically a large extra chromosomal piece of DNA that then gets inserted in the nuclear genome. Or maybe this one piece gets inserted in the nuclear genome and then it gets copied over and over and over again into this 36 to 76 copies. So far that we know of. That's just looking at the cats that we've sequenced so far. I'm sure that there'll be more variation than that when we sequence more cats. NUMTs are found in essentially every group of organisms that have been looked at. These are fig wasps. They have dozens of NUMTs in their genomes. I mentioned cetaceans, so that's dolphins and whales. And this was a relatively famous paper. I've used it in a class before too, because it's easy to have students use this type of data to create zeros and ones and then make a little data matrix. You have the NUMT or you don't have it, right? You've got this, you've got this. This species has it, but this other whale doesn't have this. And then you have a data set of zeros and ones representing characters, right? Because the presence or absence is a character for that genome representing that species. And then you can use those characters to make a phylogeny, because you can see which two whales share the most of these insertions. And those two whales are probably related to each other. And when you make that phylogeny, you end up making a phylogeny that looks just like a whale phylogeny if you based it on a whole bunch of morphological characters, or if you base it on DNA sequencing of nuclear DNA. Or even if you sequence the regular mitochondrial DNA, you'll get that phylogeny. All of that suggests that NUMTs are following a pattern of they are inserting themselves at various points in history, and then once they insert themselves, they then are passed on in a vertical fashion, and that's why we can create these nested hierarchies later on, using them as characters. All of this really applies to endogenous retroviruses. They're very, very similar in that sense. Although some would argue that NUMTs are actually a better example for common ancestry because they're less likely to move themselves later or be lost. Many of them, once they're inserted, that's where they're at. We can watch them decay. That was the NUMTs that every single person on this earth share. They're not all identical between us. They're in the same exact position in our genome, but the sequence, the code is a little bit different, and that's not surprising because as we copy our code, right, we make errors. And so they've been gradually accumulating mutations over time making them more and more different. And that's one way we can identify young NUMTs versus old ones, right? If it's really old, if this NUMT happened way back at the very beginning of the human population, you could expect them to have more differences because more generations have passed. Whereas the NUMTs that are found in a single family group, right, within like just 2 generations, grandfather, son, and grandson, those are usually identical sequences because that was a recent insertion of a mitochondrial DNA into their nucleus. This figure just is showing that there's a lot of identified, I'm going to get my pen out here. There's a lot of identified NUMTs in these different species of whales. And so there's a lot of characters one can use to make a phylogeny. And then, I'm not going to go through the details of this, but if you look at the position where the NUMTs are found, so this one is found inside of another gene. Actually, I think it's inside of an intron. And then you look at the same gene in whales versus cattle, this NUMT is found in the exact same position. So that's the other thing we find is some NUMTs are very, very old potentially. Just like endogenous retroviruses. Some of them are potentially very old, meaning they happened long ago. And they then are found in those positions in the genome. And that's the way in these whales, they're pretty consistently found in the same location or between the same genes or within the same genes between different whales that they're found in. And then interestingly, of course, many of them share the same sites with cattle, suggesting that it was in the original walking whale ancestor, right? The original land animal that gave rise to whales had these insertions, and then all whales share those. I also want to just quickly point out that plants do a tremendous amount of horizontal gene transfer, both within cells, between mitochondria and chloroplast, and even happens from chloroplast to mitochondria, mitochondria to chloroplast. There are pieces of chloroplast DNA in mitochondrial genomes of plants. The nucleus has lots of pieces of plant mitochondrial DNA and of their chloroplast DNA. And this particular example is really fascinating because, for one thing, I worked on holoparasitic plants for my postdoc. Holoparasitic plants are plants that can't do any photosynthesis. They have to derive all of their photosynthetic product from another plant. So they are parasites. They swipe their food from other plants. So they live on, and in fact, they live inside of other plants. Their tissues are mingled with other plants. And many parasites have genetic information in their own genomes, which are from their hosts. Right, so we look at their genome, we find out, you know what, this particular sequence that's in your genome, now that we sequence the whole genome, this little piece of DNA is exactly like the host DNA for that particular gene. And so it appears that DNA is being transferred from host to parasite, possibly also parasite to host. In this case, they have a holoparasitic plant that, let's get down to the nitty-gritty here, rampant horizontal gene transfer, that's HGT, has transformed the mitogenome of the parasitic angiosperm. At least 60% of the 822,000 base pair genome is acquired by horizontal gene transfer. So in this case, it's not just the host transferring information, or you could say the parasite taking the information. Depends on your perspective of exactly what the mechanism is for this. But consistently, there are many, many, many pieces of the host plants, which are all in the Fabaceae, the bean family. They are, and the parasite itself is not in the bean family, right? It's a completely different, phylogenetically distinct family of plants. The Balanopheraceae, really bizarre parasitic plants. Anyway, this particular Balanoph species has a huge amount of genes that are associated with, what did I just say? Bean plants in them. So now that we sequence genomes, and you say, like, this genome is got lots of little segments that are just like those segments over there in those other plants. And not only that, the version of the gene that would be in the mitochondria of this parasitic plant is missing, and so they have only a version from their host. And so they are more and more and more like their host. Like 60% of their mitochondrial DNA, it looks more like their host than it looks like other species of Balanophораceae. And that's really key here. Because you might say, well, how do you know that it transferred? It might just be that by chance it looks just like its host. The thing is, there's a closely related species, right, fairly closely related species of another Balanophora in that same family that has sequences that are like other family members for particular genes in the mitochondrial DNA. But this particular species doesn't have those DNA, doesn't have those sequences, even though the organism clearly is a Balanophora, right? It's not a bean plant, but its mitochondrial genome is like a bean plant. This is very similar to where I just started, which was you could do the sequence. You could get molecular markers from an individual and say, this is your mitochondrial genome. You must be related to this particular person or this particular lineage. And you could be wrong, right? Because you're actually looking at the nuclear version, which came from a different lineage. And so plants mix themselves up quite a bit. Plants are really remarkable this way. They do a fair amount of exchange of genetic information, and where we see most of it is in these parasitic plants because they're living in tight community. They are sharing a lot of information. Maybe sharing is the wrong word since they're parasites, all right? The parasites are manipulating the hosts, and they are actually emitting products, all right, from their own genome that are then influencing the expression of genes of their host. Right, so they are actually communicating, in a sense, sending RNAs out, which are probably even being translated by the host cells to manipulate them. And so it would be easy to see how the parasite could transfer to the host, but here the host is transferring also genetic information, or the parasite's taking it and then making itself more like the host, which may be a way for the parasitic plant to trick the host, all right? Right? As a parasitic infection, the host wants to try to probably eliminate that, and is probably trying to find ways to identify that it's being parasitized. And the more you can swipe pieces of DNA from your host and make them part of yourself, it kind of helps you to hide yourself from being a foreigner, right? If you actually have the same DNA or making the same proteins as your host is making. Horizontal gene transfer involves chloroplasts. Right? Horizontal gene transfer is an important driving force in the evolution of eukaryotes as well as prokaryotes. And in plants, it appears that many plants are picking up skills and abilities from other plants through this process. And not just plant to plant, it's also plants clearly have taken genes from bacteria as well and from fungi. Those are really rare events, and yet we have pretty good evidence that that has happened in multiple different groups. Evidence of mitochondrial RNA in the chloroplast genome of Convallaria kiskii and its subsequent evolution in Asparagales. So Asparagales is an order, which includes the asparagus plant, but many, many other things as well. DNA, so let's look at this. We're almost done here. DNA transfer between internal organelles such as the nucleus, mitochondria, and plastid is a well-known phenomena in plant evolution. And DNA transfer from the plastid and the mitochondria to the nucleus, from the plastid to the mitochondria, and from the nucleus to the mitochondria has been well documented in angiosperms. Right? However, evidence of the transfer of mitochondrial DNA to the plastid has only been found in 3 dicotyleds. So in this case, they're looking at mitochondrial DNA that's actually been moved to the plastid, to the chloroplast. And here what they found is that this particular species of Asparagales has a large chloroplast genome of 162,000 base pairs. It's the largest in this particular order, which includes some 5,000 or more species of plants. And the reason it's larger is because they discovered that this one species has 3,300 base pair segment of mitochondrial DNA embedded in its chloroplast genome, all right, which shows similarity with the mitochondrial gene RPL10 as a pseudogene. So this particular sequence looks like it's RPL10, which is in the ribosome, with mitochondria, but it doesn't function in the chloroplast yet. Here's this similar sequence. Further analysis reveals the mitochondrial transfer only occurred in this species in this particular family. So there are other members of the family. They looked at the things they thought were the most closely related to this particular species, and this particular segment of 3,300 base pairs is not there. All the other species have a similar size chloroplast genome. And so they think that this diverged relatively recently. For them, relatively recently is 7 million years ago. But so this species has been off on its own for a while, and it has this larger segment. So how to wrap this up? I'm not really sure. But, okay, so first I just wanted to show that transfer of genetic material from one place to another, I mean, we already knew that it happens within the nucleus itself, right? We can see that fragments of nuclear DNA get copied. There's duplications, and then I have another video coming out maybe tonight or tomorrow about the importance of duplicating DNA and then how those duplicated genes can take on new functions, right? So that's where new genetic information or novelty can arise. And we already have plenty of evidence that that occurs in the nucleus. What's lesser appreciated, and I think we see that most people are not aware that pieces of DNA are jumping around between organisms, but especially within organisms in their nuclear DNA and their mitochondrial DNA. Now, how important is this? It's really more 99.9% of the time, this is just going to insert itself in a place of no importance, and it's just going to become another fragment of DNA that then gets passed down to the descendants, right? Sometimes it's going to insert itself in a rather precarious place, right? So it's going to cause problems. So we call that a negative mutation. That's a horizontal gene transfer event which has a negative consequence. Now, obviously, if it's very negative, then natural selection will simply not allow the organism in which this occurs to survive or have offspring, and therefore that particular NUMT we won't see, right? Because it won't continue to be propagated. Some might occur and happen, and I know that there's literature on this too, but I only have so much time to get to go over all these things. You don't want this to be 3 hours. But I know there are cases where these things have jumped into regulatory regions and therefore disrupted the normal regulation, but that disruption didn't necessarily hurt the organism, but might have given it a new way of regulating. And so it actually becomes itself a regulatory element. In very rare circumstances, enough of a portion of the genome will be moved from one section to another that it becomes active and participates in its new location. And this is what is thought to have happened with the origin of mitochondria, that over time, pieces of DNA, pieces of mitochondria got shuttled and inserted into the nuclear DNA, and probably 99.9999% of the time, it's the same thing we see today, right? It had no effect, right? Or had a negative effect. But once in a while, it's going to get inserted, maybe it gets inserted into a suite of genes and gets inserted where it already has a regulatory element there, and it gets expressed. And if it gets expressed and gets exported from the nucleus and then finds its way being imported into the mitochondria, right? I'm not naive that there's a lot of different things that have to go on for you to be able to target yourself toward going into the mitochondria. But nonetheless, it's going to happen once in a while. It's going to find its way into the mitochondria. Well, once it finds its way into the mitochondria and it expresses in the nuclear genome, the mitochondrial version of it is free to decay, right? Because if that particular portion or that particular gene in the mitochondrial DNA then has mutations and it loses its function, the mitochondria still survives because it's importing that same function from the nucleus. And so that's where you would get the transfer of genetic information from the mitochondria now resides inside the nucleus. And all that has to do is happen once in a while, and then later on in a later descendant, another gene gets transferred and begins to get used in the nucleus, and another gene gets transferred and used in the nucleus, and so forth. And eventually what happens is you gradually import all that stuff there. Now, I'm remembering now, I'm remembering stuff from lectures from 10 years ago. I don't have my fingers right on it, but I'm remembering there's an example, I think maybe in rice or somewhere like that, where there's different species in which the mitochondrial genome, even in different closely related species, there's like 3 or 4 genes that are in the nuclear genome and in the mitochondrial genome, and they're both expressed. And but in another species, one is not, it has a mutation in, I think, the mitochondrial genome. And so you can see the activity in the nuclear genome, but not in the mitochondrial genome, even though the DNA is still present there. And what you're seeing there is you're just seeing the process, right? You're actually seeing the evolution of that genome. And so through looking at different species, you can see different snapshots of different examples of the process of moving from copying to the nucleus, to activating in the nucleus, to deactivating in the mitochondria, and then eventually losing the gene altogether in the mitochondria. And I believe we have examples of that probably from a single family of plants. And I'm stressing family, all right, because creationists believe that a family of organisms is a single kind of organism, and therefore might all actually be related by a common ancestry. So if you can show the complete change in the construction of the genomes within a family, you're really showing that the process happens, right? That even creationists should be able to accept that this process happens and changes genomes over time through these nuclear transfers or mitochondrial transfers. All right, that's my further thoughts. I'm sure I've forgotten a whole bunch of stuff, but we'll talk later. Maybe we'll get together and talk about some of these things sometime. All right, so hey, Sal, if you're listening, thanks. It was a really interesting discussion. I really appreciated that last night. I found, it was really stimulating, and as you can see, I found myself like, oh yeah, I know about these things, and I think I know what I'm saying here. I made some comments, but they were kind of hedged, right? Because I was like, I think I know, but I'm not going to say until I really know. And so I found myself, as you were talking, like I said, reading some papers, and then I stayed up even later, read some more papers. And this morning I got up and I couldn't help but think about these some more. And what I've just shown you is like the tip of the iceberg in terms of the things that I've seen, and I just had to sort of select out a couple examples to present to you. So great, thanks for hanging out with me. That's NUMTs, nuclear mitochondrial DNA. Hey, hit the like button, subscribe if you haven't already, and I'll be back to talk about who knows what, because I didn't know I was going to talk about this today, so I don't know what I'm going to talk about tomorrow. Talk to you later though. Bye-bye.